Hi everyone, I'm RVS Harshni, studying in Sri Venkateshwara College of Engineering. Today we are going to discuss about biology. The topic today we are going to discuss on biology is proteins. Introduction to proteins. Proteins are fundamental macromolecules essential for various biological functions. They are composed of long chains of amino acids linked by peptide bonds. Proteins play a crucial role in structure, function, and regulation within cells. The next one is structure of proteins. Proteins have four distinct structural levels: primary, secondary, tertiary, and quaternary. The primary structure is the sequence of amino acids in a polypeptide chain. Secondary structures such as alpha helix and beta sheets are stabilized by hydrogen bonds. Proteins. Proteins are classified into two main categories: globular proteins and fibrous proteins. Globular proteins like enzymes and antibodies are generally soluble in water. Fibrous proteins such as collagen and keratin provide structural support and are usually insoluble in water. Functions of proteins. Proteins serve numerous functions, including catalyzing biochemical reactions as enzymes. They are critical for cell signaling and communication through receptors and hormones. Proteins also play a vital role in immune response by acting as antibodies. These are the following functions that proteins are helpful for maintaining our human body. Amino acids and their role. Amino acids are the building blocks of proteins with 20 different types in nature. Each amino acid has a unique side chain that determines its properties and function. Essential amino acids must be obtained from the diet as the body cannot synthesize them. So the amino acids are the building blocks of proteins. So by the proteins, the amino acids are formed. The figure shows the amino acid block chain where they are non-ionized from form of amino acids and ionized form of amino acid, which is bitter ion. Synthesis of proteins. Protein synthesis occurs in two main stages: transcription and translation. During transcription, the DNA sequence is copied into message RNA. In translation, ribosomes read the mRNA and synthesize the corresponding proteins. Protein folding. Proper protein folding is crucial for functionality and is guided by molecule chaperones. Misfolded proteins can lead to diseases such as Alzheimer's and Parkinson's disease. The proteins are the three-dimensional shape that determines its biological activity and interaction. The figure shows the protein structure: primary structure, which is amino acid; secondary structure, which is A helixes; and tertiary structure, that are polypeptide chains; and quaternary structure, which are proteins. Dietary proteins. Proteins are an essential part of a balanced diet and can be found in various foods. Animal sources include meat, dairy, and eggs, while plant sources include legumes and nuts. The quality of dietary protein is often measured by its amino acid composition and digestibility. Protein analysis techniques. Several techniques are used to analyze and study proteins, including electrophoresis. Western blotting allows for the detection and quantification for specific proteins. Mass spectrometry is a powerful method for identifying proteins and their post-translational modifications. Western blotting procedure. Load and separate protein samples on SDS page. Electrophoretically transfer fraction proteins onto PVDF membrane. Block the membrane with neutral protein. Next, incubate the membrane with primary antibody specific to target the protein. Incubate the membrane with HRP labeled secondary antibody specific to primary antibody. Incubate the blot with chemilini- uh, chemiluminescent HRP substrate and expose to film. Future perspectives in protein research. Advances in proteomics are enabling a better understanding of protein functions and interactions. Research is ongoing to develop protein based therapeutics and vaccines. The study of proteins will continue to be pivotal in fields like biotechnology and medicine. These are the following references to learn more about the proteins, that is Albert's PETAL 2015 Molecular Biology of the Cell 6th edition Garland Sims. By this we can learn more about proteins which are used more in biotechnology and medicine fields. Thank you everyone. Please like, share and subscribe.